Hello viewers, welcome to a video presentation on pregnancy, infection, and autism spectrum disorder. What is autism? Autism is a mental condition used to describe disorders of brain development. Characteristics can include delayed or limited speech, hyperactivity, and impairment in social relationships. It usually develops at early stages of childhood and can even be contracted during pregnancy. Did you know that the chances of developing autism can increase dramatically from the start of development in the womb? Our genes aren't the only contributing factor in developing autism. Environmental factors that can affect the fetus can play a huge role in the development of this disorder. Infections contracted by a mother during pregnancy can create a toxic environment for the child and cause improper brain development leading to autism. Viral and bacterial infections contracted in the first and second trimesters of pregnancy, respectively, have the greatest impact on improper brain development in the baby. Studies have been carried out on mice to show evidence of this. Here's how it went. Pregnant mice were injected with a pathogen to activate its immune system. Typically, immune system responses elicit an antibody response. Think soldiers ready to fight an infection. Results showed that several of the offsprings displayed autistic-like behaviors. A cytokine in the body, known as IL-17, was produced to promote inflammation in the mother mouse, which led to an increased number of offspring displaying autism. But what is IL-17? IL-17 is a cytokine that fights infection, much like a boxer would fight his opponent in the ring. IL-17 molecules gather at the site of trauma to promote inflammation. An infected mother, IL-17's activity is high in concentration. The presence of high levels of IL-17 is directly correlated with a higher chance for the development of autism in the mother's offspring. It was further identified that the production of IL-17 as part of the mother's immune response interferes with the baby's brain development. Let's take a closer look at the mommy-baby relationship. A clinical study proved that pregnant women with high IL-17 proliferation during the first and second trimesters of fetal development delayed the production of brain cells for motor learning. The degree of communication between the fetus's central nervous system and brain can determine whether autism-like symptoms are present or not. A baby who displays these symptoms experiences slowed brain cell growth as a function of maternal inflammation. But how can we measure this? C-reactive protein is a chemical marker of inflammation that can be measured through blood testing. If CRP levels measured within trimesters 1 and 2 are high, this is associated with a higher risk of fetus development autism. There is nothing that can be done to completely avoid an infection, but remember IL-17? It can be stopped in its tracks by an inhibitor drug called consentin. An infection is not as effectively treated, but at least the chances of autism developing in the unborn baby will be decreased.